Our next speaker for today is Heejung Koo, head of the UK office at Standime. Heejung, a pleasure to have you with us today. The floor is yours.
Thanks for the introduction and thanks for having me here today. So as introduced, I'm Heejung Koo and I'm a Deputy Director of Standime UK. So for those who are not familiar with our company, Standime is an AI drug discovery company headquartered in Seoul, South Korea. And we recently arrived in the UK about 3 months ago, and we just incorporated our UK subsidiary here in Cambridge. So what we are trying to do is to create value by establishing a system that continuously supplies the first-in-class drugs. It means that we're not really concentrating on developing a couple of pipelines to the final stage, but rather we are interested in securing a smart pool of the early stage potential drug candidates. So we have been developing our AI platform for the early drug discovery stages, spanning from novel target identification to novel compound design. And now we are automating this whole procedure in the form of a workflow, starting from our AI platforms and with some mandatory experimental procedure like chemical synthesis or testing the efficacy or toxicity and something like that. And finally, getting some valuable first-in-class compounds. So this part is quite relatively controllable, hopefully fully under the control of ourselves. But this part, including the experimental part like synthesis or assays, may not be fully predictable or controllable. So definitely what we can contribute to the most is to make this part to be more efficient, highly efficient and systematic. And for this part, we are also trying to minimize the uncertainty of the wet lab procedure by providing a minimal number of, but highly diversified, potentially synthesizable compounds, so that we don't have to synthesize too many compounds, but with more high probability of getting valuable molecules. So on the whole, we're trying to systematize this whole procedure, and we started to establish this workflow from early on this year, and we have confirmed several meaningful structures from the leading project started at first, and now we are doing lead optimization for the structures. So for today, I will give you a brief introduction of this part. So how our AI platforms are doing to identify novel targets and to generate novel compounds for the novel targets. For novel target identification, we basically use a graph database constructed by ourselves, integrating the information from diverse sources. And we apply our AI algorithms on the constructed database, and we can get a list of target candidates with the evidence of the prediction. And then we apply some filters on the target list to secure the competitiveness and novelty of the target. And then finally, our internal experts will review the survived target. And now we are ready to finalize the target for further development. This is the scheme of our database incorporated in target identification platform. So it contains the information from the literature and also the public databases. It is comprised of the information such as protein-protein interactions, and pathway or biological process information, and patient transcriptome and disease gene associations, and so on. So basically, a very wide range of information that is needed to investigate the disease and target associations. And as we can see here, we have mainly these three nodes: diseases, targets, and compounds, and we have many relations connecting these nodes. And also we have some additional information surrounding these nodes to describe these nodes. So for example, symptoms for diseases, or ontologies for targets, or side effects for the compounds. Currently, we have more than 4 million relations connecting about 70,000 nodes. And here we can see how we extract information from the literature. As we separately constructed our NLP system, it receives the unstructured text as input. And first, it recognizes some meaningful entities like biological entities such as genes or diseases, or molecular physiological activities, or some relational words like this. And then it will predict the relations between the recognized entities. And by connecting the pieces of information, we can see how the genes and diseases are connected, how they are associated under the context. So this information will be integrated into this knowledge graph, and it can be also used later to investigate how well it is known about the disease and gene relations. Based on the constructed database, we use diverse features from the database and apply our AI algorithms to prioritize the targets for a query disease. We use the graph topological information and paths connecting the disease and target on the graph. I mean, the connectivity between the disease target pairs. So using this information, we can select the important paths. I mean, the paths that significantly contribute to explain the disease and target association. And we can also assign the edge weights on the graph by reflecting intrinsic network structure and also the disease-specific information. Utilizing this diverse features from the database enables us to view this problem with diverse and novel insights. And by aggregating this information, we can get a list of potential target candidates. And the targets are ranked according to the association scores calculated from these algorithms. And they are presented with the evidence of the prediction. Because we are using graph database, we can name the paths, we can find the paths that are used to calculate the association scores, and the path can work as the evidence of the prediction. And here we apply some filters to narrow down the number of the targets. And here we firstly consider the competitiveness of the target. It means that it is also related to the novelty of the target. So how many drugs are being developed with the target and how far they are progressed. So applying these filters, we can filter out the target with low competitiveness. And for the final expert review, we provide the supporting evidence from these algorithms, and we also provide some additional helpful information according to the disease area. So these are some examples of the information that we use to select the target in terms of the oncology pipelines. So this is prognosis data related to the target, and this is the cancer dependency data that we can obtain from themmap.org. After deciding which target to develop further, we can use the next platform to design the novel compound for the target. So here we again constructed a database containing the information regarding the targets and chemicals and novel scaffold. And to roughly go through the whole procedure, we first find the active hit molecules among the commercially available chemical compounds. So our platform is equipped with two different approaches according to the target structure availability. So when the target structure is available, we use kind of rescoring module based on the docking simulation. So we generate the various docking poses, and we can get the predicted energy values from for each pose. And then we further decompose the energy values to train our model. And when there's no available target structure, then we use a recommender system using the exemplar experimental data. It means that we collect compounds with lots of binding information with diverse targets, and we regard the compounds as kind of evaluation committee, and we let them evaluate how a new compound would bind to a certain target. And after finding the active hits, we change the structure of the hit molecule to secure the patentability with maintaining the overall structure. And then we optimize the novel compound by enumerating the substructures. To secure the novelty, we can explore the chemical space. So this is basically a very high dimensional latent space, conceptualizing diverse molecular properties with the structure. So a chemical compound can be encoded into a point vector in the space, and then it can be decoded back to recover its original structure. So by understanding this property, we can explore this space to find a novel structure around the fifth molecule, and then we can find some compounds with novel structure, but still maintaining the original activity of the hit molecule. And also we can use our novel scaffold database, so containing the information of the scaffold and the frequency. I mean, how often the scaffold is used to comprise the existing molecules. So we know the hit compound and we know the overall shape, and we can try diverse building blocks in the position of a certain scaffold. And then finally, we can select the structure with the scaffold of low frequency, and the resulting compound will show the similar activity with the hit compound because the overall shape is similar, but it will have higher patentability because of the novel scaffold. And for the optimization, we built an enumeration model, maintaining the core structure, but optimizing the substructure. So it works like language model. So suppose that we know a context of a sentence, and if there's a blank in the middle of the sentence, the model will try to find some suitable words for the blank. And in the same context, if we know the context of the whole structure, and if there's a blank in the structure, then this model can suggest some suitable substructure for the blank. So in this way, we can enumerate the structure, and finally, we can select the small molecules with desirable properties. So just as a quick reminder, we have been developing these modules and algorithms to systematize this whole process, and we're now running our own 17 pipelines through this workflow. I couldn't go through into specific example today, but what I want to say is that our workflow is currently going on and is being developed to improve, to shorten the time period for one iteration, and also to get the higher accuracy. So yes, that's all I prepared for today. So thank you for your attention, and I'll be happy to take questions, maybe later. Yeah.
Thank you so much, Heejung. Very, very insightful and interesting indeed.